In this second video for photosynthesis, we'll now turn our attention to see how does this process actually take place. And we'll look at the steps that occur during the light-dependent reactions and the second portion, the light-independent reactions in the overall production of sugar. And this is for C1.3, and this is the second part of this section. One of the byproducts that is essential to humans and to many other organisms is the production of oxygen during photosynthesis. And in order for oxygen to be generated and to be produced, this really begins by the absorption of some light photons that occurs at photosystem 2. Photosystem 2 is the first light absorbing complex within the process of photosynthesis, specifically the light dependent reactions, and this occurs at the pigment P680. It's called photosystem 2 even though it comes first because it was discovered second. And what happens here is two water molecules are bound together and then they get split apart, and in doing so it releases 4 electrons, 4 protons, hydrogen atoms, and then the remaining 2 oxygens are bound together to produce oxygen gas. Photolysis is the use of that light energy to be able to split the water molecules at that P680 chlorophyll pigment. This process occurs on the inner surface of the thylakoid membrane, so the membrane of the thylakoid, and those electrons that then get released from the water molecules are transferred to the reaction center, and they replace those that have been released from the P680 pigment. So some leave and then they are replaced from the water molecules. The protons, the hydrogen ions, are released into the thylakoid space to be used a little bit later, and thereby increasing the proton gradient. We'll see is necessary here shortly. And then that oxygen is released as a waste product and diffuses out of the organism, generally plants. The overall purpose of photolysis at this photosystem is to split the water, to release the protons and the electrons. Those are what's needed to be able to power the later steps of photosynthesis. The oxygen is just a waste product, thankfully for us. At the beginning of Earth's history, there was really little to no oxygen in the atmosphere, and that production of oxygen and the accumulation of it within the atmosphere occurred through this photolysis process by cyanobacteria about 2.5 to 2.3 billion years ago. The atmospheric oxygen didn't immediately accumulate because the oxygen was used up in an oxidation of iron, and we were able to see this. Iron is oxidized within the ocean, within rock, and forms iron oxide precipitate, and that led to sedimentary rock. And it produces these banded red patterns, and they're called banded iron formations, and you can actually see these. And so all of the initial oxygen that was produced went to this oxidation of iron. And then after that process, after all of the, or most of the iron was oxidized, then oxygen began to accumulate in the atmosphere, thereby allowing aerobic respiration to evolve within organisms. And so then that led to the buildup of oxygen in Earth's atmosphere. Within our image, the red and the green line represents the range of the estimates, while the time is measured in billions of years ago. And then the banded formation iron peaks at the beginning of stage 2, and then it pauses at the beginning of stage 3 after all of that iron has been oxidized. So those electrons that are generated by photosystem 2 pass to an electron carrier molecule called plastoquinone, and that's located in the thylakoid membrane, and it also accepts 2 protons from the stroma to create this the molecule called plastoquinol. Plastoquinone moves through the thylakoid membrane to this cytochrome complex, the B6F complex, and it passes those 2 electrons to the complex and then releases 2 protons into the thylakoid space. It then is converted back into plastoquinone, which returns to photosystem 2 for more electrons and protons. So it's essentially just a molecule that's carrying or ferrying those electrons from one spot to the next. At that cytochrome complex, the transfer of electrons takes place from the plastoquinol to plastocyanin, and the plastocyanin transfers electrons to the reaction center of photosystem 1. And so those electrons are just getting moved through the electron transport chain that we see here in the thylakoid membrane, somewhat similar to what we saw in cell respiration within the mitochondria. Those electrons then reach photosystem 1 and have less overall energy than what they initially started with after being excited at photosystem 2. And because that energy within them is used to pump protons, hydrogen ions, from the stroma into the thylakoid space, and this creates a proton gradient of those hydrogen ions. The concentration gradient of the protons then is essentially a store of potential energy, and then the result of this electron transport chain is this proton gradient, and it produces a high concentration of protons in the thylakoid lumen. Within the thylakoid membrane then is a protein called ATP synthase that we've seen before, and it uses the potential of those protons, their energy, to be able to create ATP. Protons move across the membrane down the concentration gradient, so they're moving from high concentration to low concentration, and when they pass through the ATP synthase, it creates ATP, similar to what we saw in the mitochondria, and it's the same process of chemiosmosis. The ATP then that is produced is released into the stroma, and that's important because that ATP is going to be used in our next step of the light-independent reactions. Cyclic phosphorylation happens only in photosystem 1, but non-cyclic phosphorylation occurs in both the photosystems 1 and 2. In cyclic phosphorylation, only ATP is produced, whereas in non-cyclic photophosphorylation, both NADPH, or reduced NADP, and ATP are produced, and those two molecules are necessary in order for the next step, the light-independent reactions, to be able to take place. So the overall purpose of the electron transport chain within this process of photosynthesis that takes place at the thylakoid membrane is really to produce reduced NADP, or NADPH, and ATP molecules in order to be able to be used for our next step. This NADPH, or reduced NADP molecule that I just mentioned, is another electron carrier, and it's similar to what we saw in cell respiration with the NAD, or the FAD molecules. The production of carbon compounds, specifically sugar, overall in the process of photosynthesis requires this electron carrier. And the reduced NADP molecule is what provides this. It's the molecule that allows this to happen. It has one extra phosphate group compared to NAD, but otherwise it's essentially identical. And this molecule is produced by photosystem 1. Light energy that's absorbed by photosystem 1 reaction center is then transferred to the P700 pigment, which serves as an electron donor. The electrons are replaced by plastocyanin, linking photosystem 2 and photosystem 1. Again, photosystem 2 is the first one that we see in the process, and photosystem 1 is second. The electron then goes through a short sequence of carriers to an enzyme called NADP reductase, and it's positioned on the stroma side of the thylakoid membrane. Two excited electrons from the P700 pigment and this electron transport chain, this short chain or sequence, then allow the conversion of NADP to become reduced NADP, which is then NADPH, and then they are thereby used to be able to, those electrons are used to create this NADP, and so then that molecule is going to be used in our second step for photosynthesis. In cyclic photophosphorylation, only ATP is produced, whereas in non-cyclic, as we mentioned, photophosphorylation, both the reduced NADP and ATP are produced. So NADP, reduced NADP, can run out when it's all converted into reduced NADP. This causes then electrons to be diverted back to the plastocyanin for ATP production, and it causes cyclic photophosphorylation. This occurs when the production of reduced NADP is not needed, or it's impossible due to there being no more NADP available for the process to be able to occur. The overall purpose of the light reactions then is to really take the light energy and turn it into chemical energy in the form of ATP and the reduced NADP. That's the function of these steps. This electron transport chain that takes place in the thylakoid membrane is to make these two molecules that are essential for our next steps, the light-independent reactions. The thylakoid is a sac-like vesicle that's found within the chloroplast, and it's used for the light-dependent reactions of photosynthesis. It absorbs light energy, and that light energy is used to split water by photolysis to be able to reduce NADP and produce ATP by chemiosmosis. Eukaryotes have two thylakoid types. One is a disc shape that's arranged in stacks called grana, and then it also has an unstacked space called that's found within the stroma. Primarily that's called the lamellae, and it forms connections between the grana thylakoid, so it forms connections between the different stacks. It's an interconnected system that has emergent properties, meaning that a property emerges as more of them are combined together, and those properties would include the ability to separate fluid inside the lumen from the fluid within the stroma to be able to create a proton gradient. It's the use of that proton gradient by ATP synthase in the thylakoid membrane to be able to produce ATP. Photosystem 2 splits water in the lumen. Plastocyanine and the cytochrome B6F complex use electron energy to pump protons from the stroma to the lumen. Plastocyanin transfers electrons from the complex, the B6F complex, to photosystem 1. Photosystem 1 absorbs light to be able to produce NADP in the stroma. And all of these things, or these components, all work together to be able to achieve the production of reduced NADP and ATP, which is essential for the light-independent reactions. The photosystem 2 and the cytochrome B6F complex are mostly in the grana. Photosystem 1 and the ATP synthase are mostly in the stroma laminae. Now that we've completed the light-dependent reactions of photosynthesis, we're going to turn our attention to the light-independent reactions, meaning no light's required. And here we'll see the carbon dioxide that's necessary for photosynthesis actually be used in order to produce sugars. And carbon dioxide is the source of carbon for all organisms that undergo the process of photosynthesis, and that carbon dioxide is used in a cycle, a metabolic pathway that's a cycle called the Calvin cycle. And so the first step of this is the carbon fixation. And this is when the carbon dioxide is converted into a carbon compound in the stroma that produces a 3-carbon compound called glycerate 3-phosphate, or G3P. And it's actually the same molecule that we saw in the process of glycolysis for cell respiration. And so this occurs through the use of a 5-carbon compound called ribulose biphosphate, or RUBP for short, and it's catalyzed by the enzyme ribulose biphosphate carboxylase oxygenase, or, because that's kind of extensive, Rubisco for short. And this enzyme Rubisco is the most common on the planet. There's about 700 billion kilograms of just this enzyme on the planet. It's extremely common because organisms that photosynthesize use it in order to fix the carbon dioxide to the RUBP molecule. And so then by taking one carbon dioxide and combining it with one RUBP, it makes a 6-carbon molecule that then is used for the later stages of this Calvin cycle. The Rubisco enzyme itself will fix 3 carbon dioxides per second, so it's actually not super efficient, but it's essential to being able to carry out this process and this first stage of carbon fixation. So again, RUBP is a 5-carbon sugar derivative that is converted eventually at the end of this process to a triose phosphate, a 3-carbon molecule, otherwise known as glyceraldehyde 3-phosphate, or G3P. And this occurs by the addition of carbon and oxygen from carbon dioxide, as well as the addition of some hydrogen that occurs by a reduction agent. The conversion uses ATP and reduced NADP from the light-dependent reactions, and it actually uses 6 of each of those per cycle of 3 carbon dioxide molecules. The ATP specifically provides energy needed to cause a reduction, and the reduced NADP provides electrons in the form of hydrogen atoms. And so then this results in the 3-carbon sugar, the triose phosphate sugar molecule, or G3P. The oxygen removed combines with hydrogen from the reduced NADP to produce water. So water is a byproduct or a waste product of this process. The conversion, this whole process of the Calvin cycle, takes place in the stroma and is independent of lights, but it's dependent on the availability of ATP and reduced NADP that's produced in those light-dependent reactions. And so the carbons that are in this G3P come from the 3 carbon dioxides that are input. So in order to start the Calvin cycle, 3 carbon dioxide molecules go in. They react with 3 RUBP molecules. And in these first 2 stages, then the output is 1 G3P molecule. In our next stage of regeneration, the remainder of all of those molecules, those G3P molecules, need to be conserved in order to regenerate the RUBP that's essential to the process. The last step or sequence of the Calvin cycle is the regeneration of this RUBP, and the G3P that was produced can be combined with another to make a hexose sugar, glucose, that eventually could be combined to produce starch. And so every time the Calvin cycle spins and 3 carbon dioxide molecules go in, 1 G3P is produced. If it happens 2 times, then those 2 G3P molecules can combine. We now have the hexose sugar, glucose, and many of those can combine to produce starch. And so that's how glucose is being made through this process, the Calvin cycle going through 2 times and using 6 overall carbon dioxides. It can continue as long as the RUBP is continually regenerated and the conversion of the 3 carbon molecule into the 5 carbon molecules. And so 1 G3P that's produced exits, the remaining 5 in one overall cycle are used to regenerate the RUBP. In order to produce that 6 carbon sugar, the cycle has to happen 2 times with 3 carbons each. And so the conversion of this 5 carbon molecule is again going to require some ATP, and it requires 1 ATP per regeneration of 1 RUBP, so 3 overall ATPs are required in total for this step. And so then this results in the production or the regeneration of the RUBP that can then combine with more carbon dioxide to restart the overall cycle. 6 turns of the Calvin cycle are needed in order to be able to produce 1 molecule of glucose. And glucose is usually converted into sucrose for transport within the plants, and then eventually converted then to starch for storage. And so 6 carbon dioxides must go in to the Calvin cycle in order to be able to produce 1 sugar molecule. The G3P that's produced, that 3 carbon sugar, can also be converted into fatty acids using enzymes of glycolysis pathway and the link reaction to produce a molecule of acetyl-CoA. Glycerol can be made from the G3P and linked to fatty acids to be able to produce triglycerides. So it is possible to be able to produce fats as well from the products of this process. It's also possible for all 20 amino acids to be synthesized in organisms that use photosynthesis by using a number of branching metabolic pathways. Nitrogen is supplied by nitrate or ammonium ions, so amino acids require nitrogen, and that's nitrate and ammonium ions provide that. And other minerals such as phosphate or sulfates can be absorbed from the soil, from the environment by the root system in plants. So ultimately it's possible for organisms that are photosynthesizing to take those products and use them to be able to create not only sugars, but also lipids and amino acids. Both the light-dependent and independent reactions are dependent on one another in order to be able to function. Light intensity is really the factor that affects which part of photosynthesis becomes the limiting reagent to the overall rate of carbon compound production. And if there's low light intensity, then ATP and reduced NADP are restricted, thereby limiting the overall conversion and creation of G3P, the 3 carbon sugar molecule. If there's high light intensity, then carbon fixation is the limiting factor, and it's the limiting step as reduced NADP becomes restricted. So this concludes our discussion of photosynthesis, this essential process to life on our planet, and concludes our discussion of the 2 linked processes of photosynthesis and respiration, and really illustrates how they go hand in hand with one another, where the inputs of one and the products of the other are linked.